Okay, hello again, everyone. As we all know, mitochondria plays a role in ME/CFS. Energy production is an issue, and we don't really understand the etiology of it. I'm very pleased today to invite Dr. Dugar to our conference. Dr. Sundeep Dugar has almost 4 decades of experience and great success in small molecule drug discovery and development in oncology, inflammation, the central nervous system, and cardiovascular and metabolic disorders. Among his many achievements, he has founded 4 different pharmaceutical companies, co-invented Zetia and Vytorin, co-authored 70+ publications and presentations, and helped to create the UC Davis Pharmaceutical Chemistry Program. 15 years ago, research in mitochondrial bioenergetics captured his interest, and he has devoted significant time and resources to this research ever since. Out of that passion came his company, BlueOakNx, and today he'll be speaking to us about mitochondria, critical for health and healing. Dr. Dugar?
Well, thank you, Lily, for giving me this opportunity, and it's really a pleasure to join you. I hope everybody can hear me and everything. So just to get started, as Lily said, today the focus will be mitochondria. Perhaps I'll tell you more than you need to know, but I think in the context of any situation, I like to tell people what I have learned, and perhaps that information will give you a complete sense of what mitochondria do and why they are so important and relevant to our health. All right, so I'll start by, let me start sharing my slide. Let me go here. So Lily, if you can just say that, let me confirm to me that you can see it, the slides, right?
Yes, we can see the slides.
Okay, perfect. All right. So, it's really in the middle of every aspect of life on planet Earth, and it's critical for health and healing. So let me just start by a little saying from one of my favorite and scientists, and also someone who I admire more, that everything is energy, and that is all there is to it. And this is not philosophy, this is physics. And I think that captures the essence of what mitochondria do in life itself. They provide the energy we need for everything we do. I mean, whether we're nodding our head, we're doing gene transcription, we're doing metabolism, or even addressing pathological insults. Mitochondria are in the middle of it all. There is not an operation that your body does that does not involve the mitochondria. And it's true for every species on planet Earth. I mean, from plants to animals and fish and everybody. So how did mitochondria come about? So it was a theory proposed by Dr. Margulis in the 60s about the role of generation of mitochondria through a process of endosymbiosis, when an ancient ancestral eukaryotic cell was able to phagocytose a bacteria body, an aerobic bacterium, creating an endosymbiosis where the bacterium, instead of being digested, became part of this mitochondria, of this ancient eukaryotic cell. In short, the product of this mitochondria started to provide the energy for the eukaryotic cell, and there was a symbiotic relationship that was created. And that is fundamentally how it has been proposed that mitochondria became part of our system. And this really puts a new parallel concept on the evolutionary theory. And endosymbiosis, it goes beyond the gene. It really is central in terms of evolution based on metabolism and energy as a central organizing feature of life. Because without the mitochondria, your cells can't function. If you take this mitochondria out of a cell, the cell cannot make mitochondria by itself. Mitochondria are present and they make more of themselves. And they are the ones that then sense cellular issues and response. And so this part of mitochondrial process then focuses on biochemical networks rather than genes, on phenotypic interactions rather than informational inheritance, which is genetic inheritance. And so endosymbiotic views of evolution, hence, are valuable for expanding evolutionary explanations. Of course, it's not an alternative to standard evolutionary theory. It does not make the genetics irrelevant. It just positions the relevancy of bioenergetics in our evolutionary process. Because after all, energy is the primal force and the basic component of the universe. I mean, what sun does is provides the energy we need to survive on planet Earth. That's the sole purpose. Light and heat is what the sun provides us. So, hang on. Again, to reiterate, and they're critical. So they are present in every cell of the body, except for the red blood cell. Every cell in your body has mitochondria. So you can imagine that at the point of any pathological insult, when the cell is under stress, the mitochondria provide the energy for that cell to respond to that stress. And that goes through whether it is functional stress, such as exercise, or it is pathological, such as, say, MACFS. Each provides a stress to the cell that initiates the mitochondrial processes within your body. Okay, let's go through some interesting facts about the mitochondria. As I said, it's the only component in your cell that can make more of itself when it's needed. And the basic way you make more mitochondria is exercise. And I'll come back to that a little bit later because that is germane to the discussion we're having about ME/CFS. They do get broken, so they do get affected by cellular stress, but they fix themselves. And when you have too much mitochondria in a cell that you're not using, they get rid of themselves, which again gets to the idea that if you're not exercising or if you're not physically active or otherwise, you lose more mitochondria because the cells do not maintain organelles or systems that are not in use. Their sole function or primary function is to take in nutrients and turn it into energy and fuels all cellular activity. We typically have about 100,000 trillion mitochondria in our body, occupying about 25% of cell volume. Each one of us uses about 10 billion ATP per day. And healthy person produces their body weight of ATP every day. So if I were to take the amount of ATP you make every day out of your body and weigh it, that would run up about 150 to 160 pounds every day. And what is fascinating is that ATP cannot be stored. So it must be produced every second of every day in every cell and tissue that is alive in your body. And then when you move and you exercise, the tissue, that tissue that you're exercising initiates its own mitochondrial biogenesis to make more mitochondria to meet the energy demand you're putting on it. And it's happening very, very fast and in an instant. But at the level of about 21, age of 21, you peak, your mitochondrial levels peak, because that is when your growth stops. From then on, you lose about 10 to 15% per decade. That's called aging. So let's put this in context. After age 21, you're automatically losing your mitochondria. When you have pathological stress, you lose more mitochondria because you're using mitochondria to address this pathological stress. And then if you don't exercise or cannot exercise on top of that, you lose even more mitochondria. So this becomes the spiral that you go down, and you have the consequences are the phenotypic consequences you normally see, even in diseases such as ME/CFS. And so besides that, you can also lose mitochondria through your environment and through your diet. So such as glucose, high sugar. So yes, are the odds stacked against us in a way? Yeah, in a way. But human evolution has allowed us to live reasonably well. And so many ways to lose mitochondria, but still there's one way to make more mitochondria, and that is exercise. And that's the part that we've been looking at. What is it that happens when you exercise to make more mitochondria? And it's a question that when I started in this, though well known, I asked all the experts in the area, and they could not tell me what was the mechanism or the biochemical pathway that happens when you exercise to make more mitochondria. So, recapitulating everything, if so, it is central to cellular function. They provide energy. That almost every pathology affects the mitochondria is foundational truth in biology. And this function can cause primary. That is, if your mitochondrial DNA it itself has a mutation, or contribute to progression of disease, which is secondary. That means as you lose mitochondria, you start to see the comorbidities, such as chronic illnesses like diabetes starts with high insulin, high sugar, what slowly leads to background inflammation, cardiovascular disease, kidney disease, etc., etc. So those are the progression of the disease. And then when you exercise, you make more mitochondria. So on the left here, you make more mitochondria. You create improved function by doing cristae remodeling. And cristae is the place where the cell mitochondria make ATP. You create longer, healthier mitochondria through mitochondrial fusion, which increases mitochondrial respiration. And also, it induces mitophagy, where if there's any broken mitochondria are removed from the cell. So it's like a house cleaning. And so when a body has sufficient mitochondria, it does all of this very effectively. And that's what exercise does. When the body doesn't have enough mitochondria, it starts to make choices and may not induce mitophagy or not induce enough fusion because it has to make choices as to what the bioenergetic needs to be used for. So it's simply a currency exchange. So if you have enough of it, you can have a 4-course meal. If you don't have enough of it, you just get the starter, for example. So let's get into the exercise part. So the question to my mind was that one of the first things happen when you exercise is you make ROS, reactive oxygen species. Now, this is different than when you make ROS when you have a dysfunctional cell or a dysfunctional mitochondria. So dysfunctional mitochondria can also make ROS, and excessive ROS can be damaging. But at the right time, at the right amount of ROS, it's pathologically good because it really gives you the adaptive responses to generate antioxidant defenses, higher oxidative tolerance, etc. So the theory has been ROS is bad for you. Hence, we all take antioxidants. The idea here is reactive oxygen species suppress it. But this runs contrary to the fact that exercise which induces mitochondrial biogenesis, also produces ROS. And there's a disconnect here. So often people will tell you that here's mitochondrial respiration, generates ROS, and causes all this damage. But that's not true when you exercise. So where is the difference here? And the difference is on the level. So when you make ROS and you stay within the beneficial zone of exercise, it's good for you. When you have too much of it or too little of it, it's not good for you. Too much causes a lot of damage. Too little doesn't induce the mitochondrial biogenesis processes. So the answer is not a black and white answer. The answer is that a limited amount of ROS is good for you. And either too little or too much is bad for you. So let's ask the question, why is this limited amount of ROS good for you? What happened? So the question in my mind came that exercise induces ROS. Hence, it has to have some positive part because cells are not gonna make a damaging biochemical when you exercise, because we know exercise is good for you. So if you're making ROS, what happens? So let's go to the next step. Exercise induces ROS, potentially in this beneficial zone. But then what happens when you induce ROS? Now this it's a complicated slide, but it's I'll simple it down very easily. Mitochondria start the synthesis of all steroids, okay? So they control two key steps in steroid synthesis. The first and one of the last. These are enzymes that get activated by ROS. So, interestingly, when you exercise, you make ROS. ROS can activate these two enzymes that sit in the mitochondria. So the question comes in, if that is the case, what happens when these two enzymes are activated in the mitochondria where all these steroids work? Well, what happens is that the product of this would be a new steroid. So forget all the other steroidogenesis that is happening in the adrenal gland. But post-exercise, when the mitochondria are activated, and these two enzymes in the mitochondria are activated, just these two enzymes would produce this specific steroid. So the question is that what's happening? So I went to PubMed and found nothing on this steroid at all. There was no report of it in terms of its pharmacology. I said, that's strange, because here's the putative biosynthetic pathway. And I'm not going to go into the details of it, but here's exercise. ROS goes to peroxide, activates the two enzymes, and then makes this steroid. But then another thing can happen, which is when you exercise, you change the ratio of NAD plus to NADH, it increases it, which then becomes an oxidant and activates 3-beta-hydroxysteroid dehydrogenase and converts this to this. The only difference is this part of the molecule has changed to a ketone. So from a biosynthetic and exercise part of it, the question became, is this the whole process? And since there was nothing known about this, I thought perhaps this could be the entire story. Anyhow, so here's hydroxypregnenolone, and here's the progesterone. And this slide deck tells the story. What we found was that this first steroid, the green one, exists in the mitochondria. We were the first ones to identify it and study its biology and pharmacology. And it's produced when you induce exercise. And it's released during increased oxidative demand through exercise, and we have shown that. We've also shown that it induces mitochondrial biogenesis at picomolar concentration. It shifts the mitochondrial fuel preference to lipids, and then also does downstream expression of regenerative proteins in muscle and neural tissue. And we've shown all of this. And I'm giving you about 6 years of work in about 2 minutes of presentation. But what I'm trying to tell you is that this hypothesis, as it turns out, we have this confirmation that this is happening because we have also detected it in humans. And in the humans, in the blood, it is the oxidized version. And here are these are scientists in my lab, and we looked at their blood, and it exists in all of them between 17 to 35 picogram per mil. So interestingly, this also explains why exercise is good for you, and it benefits your entire body, not just the tissue that is exercising. Because the tissue that is exercising gets the benefit through the autocrine pathway because it's the tissue that is making it. But the general effect on your whole body is through the paracrine effect of this steroid, which is released into the blood. So in summary, exercise makes the ROS. ROS activates the two enzymes produce the pregnenolone, and then it works within the cell, and it is released into the entire circulatory system as this oxidized version, which does the same pharmacology, okay? So having established that, we said, okay, this is great, but here's some data to confirm what I just said, that if you look at the formation of new mitochondria as we measured in the increasing levels of electron transport chain protein when we treat cells with 11-beta-hydroxypregnenolone, you can see the EC50 is very, very low. This is picomolar range, and ETC1 is approaching femtomolar activity. So this is the confirmation that it is inducing mitochondrial biogenesis, but also these downstream transcriptional proteins that you would expect to go up also go up. So the path is not just the creation of the mitochondria, but the subsequent improved transcriptional effects of hydroxypregnenolone. So let's step back and say, okay, bioenergetics of the mitochondria are closely associated with the production of the steroid, which is associated with exercise. Now looking at ME/CFS, okay, and these are statistics you guys know, so I don't have to tell you. And it is a global health issue. So what triggers it? Many cases of ME/CFS begin after an infection, suggesting an abnormal immune response to an infection may be a trigger that leads to chronic state of inflammation. May not be the only one, but perhaps one of the primary cases. And the immune system overreaction condition can be seen as a neuroinflammatory disease where the immune system has an abnormal prolonged response. And this is the clinical presentation. Okay, this is based, what it is. So all these 8 and perhaps more clinical presentation result in ME/CFS. Interestingly, mitochondria are affected under all these conditions. So what is that central role of mitochondria now in these ME/CFS patients? Due to the triggering and then subsequent clinical presentation that leads to these phenotypes in ME/CFS patients. Now, mitochondrial dysfunction, as Lily mentioned early on, is there in MECF patients. Sometimes they see higher ATP levels, but that could be from non-mitochondrial sources. So here, a qualifier. Mitochondria will make about 90 to 95% of your ATP, but there are alternate ATP synthetic pathways, but they're very minor. But those higher ATP levels can be found in some instances of MECF patients, perhaps as a compensatory phenomenon. Now, there's also evidence of increased free radical and oxidative damage. Muscle biopsies showing severe deletions of genes in mitochondrial DNA related to energy production. And then, of course, reduced levels of carnitine and N-acetylcarnitine, which are essential for mitochondrial function. This is some of the background in MECFS patients. The interesting part is that the heterogeneity of the clinical phenotype, I only gave you 8 in the previous slide, makes it hard to get a single unified picture and relevance of mitochondria. And the issue is that we, in our way of a reductionist process, try to associate the phenotype to a singular pathway. And hence, we call it heterogeneity. So whether you have neurological issues because of MECFS, or do you have skeletal muscle issues with MECFS, that is a clinical presentation and perhaps representative of your genotype. And what we, in a reductionist form, say that if you have skeletal muscle dysfunction, there may not be any association with the patient that has neurological issues. But perhaps there is, because both of those conditions have a singular data or singular point, which is the mitochondria. So the mitochondria are damaged. It's just that in different patients, the consequence of that is different, causing heterogeneity. So something to keep in mind. Hence, considering the clinical phenotype through an N of 1 perspective may provide a more direct association with the mitochondria. So if we so in the previous slide, as I mentioned, you can take pain and headache and light, noise, or touch sensitivity. And instead of going through the singular pathway of causalities of this, if we assume that the central point is the mitochondria are in the center of it, and these are simply consequences part of different patients, would tend to take this heterogeneity out of that. And perhaps that's how we should look at patients with MECFS. So targeting energy metabolism presents a potential therapeutic strategy. And there is some evidence where combinations of natural supplements such as L-carnitine, CoQ10, lipoic acid have shown help reduce fatigue in patients. And they're all known to be mitochondrial supplements. But they don't promote generation of new mitochondria. So there's a difference. It's almost that you're providing the fuel, but if you don't have enough mitochondria, the consequence or the benefit of that fuel may be limited. Okay. So here's the idea that exercise intolerance and slow recovery from fatigue after physical exertion are hallmarks of symptoms of MECF. And it could be because of impaired mitochondrial function. There's a slow regeneration of ATP, slow oxidative metabolism, and increased stress in the muscle. So consequences of dysfunctional mitochondria can also limit function of virtually every system in the body. Okay, and I list them here. And then PEM, which is a key symptom of MECFS, with neuroimmune features, could be mediated in part by the mitochondrial innate immune signaling mechanism involved in skeletal muscle adaptation to exercise. Because the connection of the muscle to the neuromuscular junctions to the neurological features, to the neuronal system is very clear. So what if in MECFS patients, the exercise intolerance is not an indication of aggravation of the disease, but a consequence of poor tolerance or recovery from the exercise. Okay, so they for them it is a very clear thing, and some evidence is starting to is there. For example, if you look at peripheral blood mononuclear cells in mitochondrial function in older patients, patients with fatigue exhibited more functional limitations, more depressive symptoms, and lower PBMC mitochondrial respiration. And the same thing happens in patients with ME/CFS, also showed altered mitochondrial metabolism with a decreased ability to fulfill cellular energy demands. And this is with ME/CFS patients in PBMC. So again, just reiterating that mitochondrial dysfunction contributes to fatigue symptoms, and improving mitochondrial function can increase the amount of ATP energy available for use by cells. The conundrum is, how do you do it? Because, as I've told you, that exercise is the way to do it. And I'm not going to go through this again, but simply says exercise effect on the mitochondria, exercise that benefit mitochondria, benefits of exercise for mitochondrial health. All this thing is very well known. I'm going to skip this slide because I think we've touched on this a little bit. And this is systemic effects of exercise. And again, it's part of reiterating. And this is a consequence of improved mitochondrial function. The other thing that exercise does is releases myokines. Myokines are small peptides and proteins, and they communicate between muscles and other organs. So I told you about the steroid. The steroid's role is to simply make more mitochondria. But once you have mitochondria, the mitochondria do these secondary transcriptional things or release these biochemicals that improve muscle growth and repair, metabolic regulation, endocrine function, and brain health. So this really comes to the conundrum of it all. Exercise does all these things, makes more mitochondria, secondary transcriptional processes, brain health, skeletal muscle health, metabolic health, everything. And one of the first things that ME/CFS patients cannot do is exercise, because that can be harmful. Now, people can say you can do light, moderate things, but you just don't know when you will hit PEM. So you've got a problem, because how do you calibrate your exercise that could be beneficial without tipping over into PEM? Okay, so this is what I've already covered, and we need to avoid overexertion, as everyday activities can lead to a crash, and most patients have no idea what to do except, they're advised with gentle birthing. And it's hard to follow. So let's go back to this whole idea of good ROS and bad ROS. What we have to do is get here. ME/CF patients probably are here, where they have excessive ROS. And that ROS is damaging the mitochondria even further. And how do you bring them back to this particular level? Okay, I'm gonna tell you about this again. And what we found was the best way to do is to perhaps, let me go back here for a second. So if you cannot make the ROS to do this, or you have excessive ROS, and you cannot exercise to do any of those things, and ROS makes this, can we use this as a supplement? It would prevent you from having the need of the exercise, but get the benefit of the exercise. and perhaps it's the solution of the conundrum. But as I mentioned, these are steroids. They're very, very potent and pharmaceutically not validated. What we found is that epicatechin is a structural homologue of these steroids and shares close pharmacological properties with both. Okay? So instead of giving the steroids, which are very active, hard to dose, and do not have, we don't have assessments of its pharmacology and biology in human dosed as a therapeutic, what we found was that epicatechin is a structural homologue. And epicatechin is found in green tea and in cacao. And this has been the study that I've been doing. We've done multiple clinical studies, published them. We're doing another 6 or 8 clinical studies in various places, in various indications. Because the idea is simply that give your body the signal that it is exercising without you having to exercise, and then have the benefit of the downstream processes of creating new mitochondria and all the transcriptional events that needs to happen so that you can get the benefits of exercise. Okay, so here's the data again, like I showed before. Epicatechin again increases mitochondrial electron transport chains, and the EC50 is not as potent, but is still significantly potent, right? So it's about 1,000 to 1,500-fold less active, but as an intervention, still significantly active. So one of the things I want to show you was a study of epicatechin in exercise. And if you look at it, these are 4 groups, but you just have to focus on this group and compare to this group. So these are animals that only got water. These are animals that got epicatechin at about 1 milligram per kilogram dosing. And if you see from day 0 to day 15, these guys did not change much of their duration or their speed or their distance or the amount of work that they were putting in. But if you look at what happened to the epicatechin group, their duration of exercise went up. They didn't get tired as much. Their speed went up. They were running and exercising faster. Their distance running went up significantly. So they were running not only longer, they were running faster, and they were covering a longer distance. And then the work output had increased from 377 joules to 533 joules. What I'm trying to tell you is that in a normal, healthy animal, but there are various other studies we have shown, even in the clinic, non-ME/CFS patients, these are patients with diabetic cardiomyopathy or Becker's muscular dystrophy, that have shown similar enhancement of muscle function, heart function, and inflammation. So what I'm trying to tell you here is that epicatechin does have an effect on your physical activity if you take it with it. So I'm coming to the end of my presentation, and then I'll leave time for questions and answers. That 2013, early part of my research, mitochondria was one of the components of all the other components that was relevant to human health and health span. I think in 2025, we can say mitochondria are in the middle of it all because they control all of these other parts, from chronic inflammation to stem cells. And while I mention these specific changes, you can imagine that these specific cellular changes have a significant physiological changes in humans and in the phenotypic changes that they cause in humans. So at the end, I want to say it's all about managing and having functional productive mitochondria and good levels of mitochondria can only help benefit. It is not a therapeutic. It is something that can benefit the clinical symptoms and clinical presentations of diseases. We have shown it in multiple diseases, diverse set of diseases. And my sense is that epicatechin or mitochondrial role in ME/CFS can be beneficial. It's just that up until now, there was no way to have the benefit of exercise be given to patients without them having to exercise. And perhaps with supplements and of our understanding of biogenesis pathway, there's a potential of providing help to patients by at least inducing the positive outcomes of exercise without them having to exercise. So I'll stop there and happy to take questions.
What a great talk, Dr. Dugar. I especially liked how you integrated what we know about ME/CFS into your talk. And I learned a few things. I did not know that ATP decayed so quickly and that the mitochondria could reproduce itself, but we still have a lot of barriers to it, reproducing itself and repairing itself. I'm going to turn it over to Dr. Nakul to help with Q&A. I have to go to take care of another session, but thank you.
Thank you, Lily, so much.
Thank you very much again for your great presentation, Dr. Dugar. Very, very enlightening, very, very stimulating. We have a few questions here, and I'll start with the first one from Philippe Tournesac. How do you explain that some patients with coenzyme Q10 deficiency in the blood are aggravated with coenzyme Q10 treatment?
I mean, I don't know if I have an answer for that question, okay? Because CoQ10 is a stimulator. The only speculation I can make is that when you do a mitochondrial not a mitochondrial biogenesis promoter, mitochondrial activator, in the presence of a deficit mitochondria, okay, can you initiate more oxidative damage? So what do I mean by that? That what is the role of CoQ10? CoQ10 plays a role in the electron transport chain that creates what we call the proton motive force. It creates the potential, okay? That's all it does. CoQ10 belongs in the electron transport chain to create the potential. And the question comes in is that in cases, and this is hard to study preclinically, but in some cases in the clinical presentation, the supplementation of CoQ10, when you do not have decent number of functioning mitochondria, can induce these potentials, which can create more oxidative damage. Because remember, what is the potential? Meaning the mitochondria is putting protons out into the membrane, inner membrane, I mean, the intermembrane space, and it's acidic, very acidic. And if that is not well controlled by the mitochondria or how you uses it, it can be reasonably unhealthy for the cell. See what I'm saying? Mm-hmm. So it has to be contained. So that's the closest I can come to giving you a speculative answer, but it's hard for us to recapitulate that in a study to understand preclinically how that could be happening.
Dr. Dugar, I have another question. If it makes a difference what type of exercise and let's say aerobic exercise will stimulate mitochondria production better, or it can be any type of exercise?
any type of exercise, aerobic, but always, as always, a combination is better, right? So you know that if you do aerobic exercise, your metabolism goes up for the next couple of hours, okay? But if you do strength training, right, resistance training, your metabolism goes up for the next 4 to 6 hours, okay? So my recommendation is both. But any exercise is better than no exercise.
Right, thank you. I have another question from Robert Fredericks. We agree with mitochondrial dysfunction being at the source of the pathology. Do you have any thoughts on what the systemic mediators of this behavior response are?
So if you believe, then the mitochondrial dysfunction will not be localized to a certain area. Mitochondria, even within the tissue and systemically, have a coordinated process, okay? So remember, the steroid I speak of, you make it every day because mitochondria have a half-life, okay? So every 28 days or so, they turn over. So you have to make, the mitochondria have to make their own anyhow, even if you're not exercising, just to keep enough in the cell for the cell to be alive. Because if there is no mitochondria in the cell will die, okay? The cell cannot live without the mitochondria. So the cell naturally makes some mitochondria. They are not making enough. This turnover is not good enough. So the mitochondria really provides you a more centralized hypothesis that the systemic dysfunction can still be now associated with the mitochondrial dysfunction in each of those tissues. Now, given our phenotype, we may have gradations of dysfunction, that in some cases it may be more skeletal muscle, in some cases it may be more metabolic, etc. That's a difference, but that does not preclude the idea that every tissue bed will have it because every tissue bed has mitochondria.
Right. So thanks for that. So Teresa Clark has a question. I'll try to pronounce correctly. What is the difference between epicatechin and epigallocatechin gallate?
Gallate, yeah. Big difference. So there are two distinct molecules. From my work and work of my colleagues and others, we have shown that it's only epicatechin that has this effect, okay? Epigallocatechin gallate does not, epigallocatechin does not, catechin does not, quercetin does not. So it's a very specific structural feature of epicatechin that allows it to have this effect. Let me add another part to that. And what we have learned is that there is a sweet spot of how much you should take, okay? Because it's the same thing, because this is direct related to exercise. You cannot exercise for 8 hours a day. You cannot exercise all the time. You only need to exercise, and you can only exercise for so long because your body will stop, right? So epicatechin, since it's mimicking exercise, the effects of exercise, you only need a small amount of it that would replicate the duration of a small amount of exercise every day. So if you take too much of it, it stops working. Okay? The other part is, that's why when we researched it, I was very clear in understanding other flavonols that are like this, and they either did not work, but if they were present, they interfered with the activity of epicatechin. So you need the right dose, and you need absolutely the right purity.
That's really challenging in practice. So I'm glad that you're doing lots of research on that to guide the right amount of use. So
Yeah.
just one question. I mean, of course, when you have the epicatechin, the evidence is that you will improve the distance that you can go, certainly in animals, but I understand in humans as well. It'll improve the performance, so people will exercise more. So how is there any risk by doing that people with ME might have more PEM just by using the medication or the supplement and then being able to exercise more? And how do you balance this in relation to a potential risk of PEM? Or in other words, is it protecting against PEM at the same time?
So I think so first one, Luis, some of that question we still have to answer, okay? Mm-hmm. Because there is no preclinical model to address chronic fatigue syndrome, right? So, so you're absolutely correct. Now, but let's focus on the idea of why does PEM happen? Part of the PEM is that your body is so devoid of energy that the moment you stress it, it goes to exhaustion because it doesn't have any baseline amount of energy to begin with. Okay? So if you keep that in mind, when you supplement the mitochondria and it goes up, okay, now you may be able to say walk up the stairs and walk better, okay? Mm-hmm. But fine. So your mitochondrial levels are starting to go up, all right? You're climbing up. So the heart of your question is, can you get too much mitochondria at that point? Correct? Mm-hmm. And that's where hormesis comes in. Remember, cells have an hormesis. So I just mentioned to you, if you take too much epicatechin, it stops working. Okay? So if your mitochondrial level is getting towards normal and you take epicatechin, there will be a time when the internal signal of hormesis will stop it. The control resides within your own cell, that you cannot have excessive mitochondria. And that's what happens. So typically, if your mitochondria are functioning at, say, less than 50-40% capacity, the cells start to get rid of the mitochondria because it costs cell energy to maintain that mitochondria, so it gets rid of it. So that's the theory hypothesis, that you should not tip into PEM because you do not have a deficit of energy. Okay, so you're going up. But do you induce mitochondrial toxicity? My hypothesis is known because that is now under the hormetic control of the cell itself. And the cell will say, you've exercised enough. If you take epicatechin, it does nothing. Yeah? Mm-hmm. So that's why the human body is fascinating, isn't it? Because it knows it needs energy, but too much is not good for it, right? Like if you take a 110 volt thing and put it into 220 volt, it'll go crazy, right? So energy is very well balanced in our system.
Perfect. I'll just move on to the next question.
Sure.
There are quite a few here, and we only have 5 minutes. Yeah. Let's try to cover as many as we can.
Sure.
Some Keating ask, curious about any red blood cell thoughts, given some known red blood cells abnormalities in ME/CFS, plus the fact that red blood cells lack mitochondria.
Yeah. So the erythrocyte carry a lot of glucose. They're glucose energy transporters. So, they don't have mitochondria. I do not know the causality of RBC dysfunction within ME/CFS patients. Now, one of the things that red blood cells do is carry oxygen, right? So they have heme proteins, they carry a lot of oxygen. If there is not enough oxygen being used by the tissue bed, do you have oxidative damage of the red blood cell because the oxygen does not have a utility in that body? I don't know the answer to that question, Luis, but these are the questions I ask and I try to figure out what is going on. But a good way to understand is to really look at erythrocyte, morphologies in these patients and understand whether upping the mitochondrial function and increasing oxygen consumption by the cells helps with the morphology of the red blood cells. But I don't know the exact answer to that. But those are my thoughts on scientific terms.
Thanks for that. That's helpful. Mark Kamenzind is asking, is green tea or cacao or cocoa or specific supplements most practical to promote mitochondrial synthesis for severe ME CFS patients?
Well, as I said, the problem is that they contain epicatechin, but they contain a lot of the other flavonols and other materials that probably are not good for ME CFS patients. What we have learned is that if you just take extracts that are not pure, see, my example, Luis, would be that if you have a headache, you take aspirin. You don't have to go chew the bark of a willow tree, right? Because the willow tree also has aspirin in it, salicylic acid, right? But you don't do that because for an intervention or a supplement that has a very clear mechanistic path, we have to do that. It's the same thing, the food we eat, we clean it, we make sure that, you just take the carrot, not everything else, right? So they are good, but they are not, we have shown also in our studies, they don't know, first of all, you don't know how much you're getting, what are the impurities you're getting, and as I said to you, purity, dose, etc. And the final thing I will tell you also that epicatechin is reasonably unstable. So when you treat it with air and hot liquids, it will polymerize and oxidize. Okay.
I'll just go for a final question because it's time to close very quickly. What about, how about magnesium?
No, I think magnesium is a good part. I'm not sure it has any relationship to mitochondria itself. I think magnesium is a critical supplement, heavy metal that you can take like any of the other supplements, but we have not been able to see a real clear association between magnesium and mitochondria.
Okay, so thanks very much indeed, Sundeep. It's a really, really excellent presentation. I'm sorry that there are a few questions that we didn't have time to address. So, but really delighted with your presence. The next activity will be the workshops too. And so there are three options, of course. One in track 1 is using occupational therapy to pace in order to achieve independence. Track 2 is understanding ME, CFS, or scoping review, illness severity and burden. And track 3 is explaining ME, CFS, simple and visual models for a complex illness. So for all the attendants, please log off of track 4 and go to either track 1, 2, or 3 according to your preference of workshops. Thank you very much.
Thank you, everybody. Bye-bye.
Bye-bye now.
Bye.
Okay, thank you.
Thank you very much. Yes, I'm going to close it to everybody. So
yeah, do we tend to, I think are we supposed to dismiss those, or I think I've done that? Okay.
Yeah, I will just, add to all. Thank you very much.
Okay, I'll see you somewhere else now.